I want to start by thanking everybody for coming and saying how deeply honored I am to be able to give this lecture today. And I want to tell a little bit about the journey of where I've gone and how the science hit as we went there. So I came to the University of Minnesota actually in 2000, trained on the one side as a computer scientist and on the other side as a neurophysiologist, where I learned to study a certain kind of cell in the hippocampus, a brain structure involved in learning and memory. And that cell is called a place cell. And basically, we could record from rats running back and forth on linear tracks. And so as the rat runs out and back, you can see that this cell, shown by these dots here, that spiked only in a single location. And so those cells represent location within the environment. However, when I got to the U, I knew that hippocampus was a very crowded place. And so my first graduate student first graduate student being Neil Schmitzer-Torbert, Neil and I said, let's go elsewhere. Let's take all this technology that had been developed, and let's actually go and try to record from a different brain structure called the dorsal striatum, which we knew was involved in procedures. And so we trained a rat to learn a procedure of running through a sequence of choices and then turning left for food or right for food. And we started with a spatial task because we knew hippocampus was spatial. But then we also did a non-spatial task where the rat had to count to 5. So if the rat is here, it has to go 1, 2, 3, 4, 5, and then it would get food. But if it was here, it would go 1, 2, 3, 4, 5, and now it would get food. So you can think of this task as being in 5/4 time. So of course, we call it the Take 5 task. And there's a couple of jazz fans in the audience. What we discovered was that unlike the hippocampus, the dorsal striatum only represented space if space told you what to do. If space was not informative, it didn't tell you anything about space. And so I started writing my first talk, probably about 2002, 2003. And the talk was obviously going to go, in the dorsal striatum, we see this on decision tasks, and in the hippocampus. And I realized nobody had ever run an experiment looking at hippocampus, even this crowded world of hippocampus. Nobody had ever run hippocampus on a decision task. And so I convinced actually my third graduate student, Adam Johnson, to go and do that, to run hippocampus, record from hippocampus on a decision task. And I love this story, speaking of serendipity, because Adam spent about 2 years whining that he was just doing what Neil did, and there was nothing interesting here because everybody knows what hippocampus is doing. And then one day, Adam comes into my office and says, Dave, my rats are doing mental time travel. To say I called him crazy would be polite. I think I said we don't use words like that in public. There's no way that's right. What he had actually observed was that when the rat came to the choice point, those hippocampal cells were sweeping ahead of the rat alternately back and forth to the goals the rat was going to. And the reason he had done that is because he and my second graduate student, Jaden Jackson, and I had developed a technology that could, in fact, read rats' minds and listen to their dreams. And I like this term because this is not metaphor. I mean this literally. How do we get there? So we call this the decoding loop. For us, we have some neural signal. For us, it's going to be recordings from these electrodes placed into the brains of rats. But it could be fMRI or EEG or any other neural signal. We have a behavior here. For us, the position on the rat on this cylinder. But again, it could be any sort of behavioral signal or even any information in the world. We can talk about how those are encoded. And so each of these circles is the firing of a cell, a single cell, as the rat runs around this environment. And this is 93 cells recorded simultaneously from the hippocampus of a rat. And you can see this cell, for example, likes this kind of southern half. This cell likes the center. This cell kind of likes that, southwest corner, et cetera. And now, if we know this, and we observe that a subset of these cells are active, we can decode the information. We can basically say what position in the environment is consistent with these cells being active. Now, there's a third piece to this, which was a real key insight that Jaden and Adam had, which was what we call self-consistency, which is basically if I know where the rat is, I know this decoding, and I know these tuning curves, I should be able to repredict the spike trains. And if we can do this, I would argue we understand something about what's going on. The problem here is what happens if we decode to somewhere the rat's not? So for example, if we have a rat running around a circle, take the rat off the circle, put it on a flower pot. Yes, that is a towel in a flower pot in a trash can. You know, this is, you use what you need to. The rat would normally be asleep. This is not asleep. He's posing for us. But a rat would be asleep. And if we record from those cells, what we'd observe is not firing about the flower pot, but rather firing about the track. And that's what we're showing here. Red is high decoding, blue is low, and each loop here is a loop of the track. And I hope you can see a sequence running along the path of the animal. This is the animal dreaming about what it did. And so as I said, what Adam had seen was that the rat was planning, was running these sequences, imagining options ahead of it. And one of my themes today is going to be the amazing collaborative possibilities and the mentorship that I've had over the years at the U. And one of those mentors was a guy named Bruce Overmyer, who is a psychology professor. And we had been interacting with Bruce through the Center for Cognitive Science. And Bruce said, oh, you should go look at the old Tolman data. Okay, what old Tolman data? Well, it turns out that in the 1930s, Evelyn Gentry, and I want to make sure I give Evelyn Gentry credit, because she's apparently the person who discovered it. And this is an abstract, and she's not on the paper. And Munzinger later says, I should have had her on the paper. I don't think it's a woman in science thing. I think it's a, she had left the lab thing. But nevertheless, giving women in science in 1931 credit, it was Evelyn Gentry. But Munzinger, Gentry, and Tolman in the 30s and 40s had observed this behavior they called vicarious trial and error. And they thought, they claimed, the rat was imagining the future. This is a behavior, it's easiest just to see it. So I'm going to show you one lap of a rat. Let's see if it plays. Did I click it? Let's try that again. There we go. And so watch what happens when the rat gets to the choice point. Anthropomorphically, it looks like the rat is unsure of where to go and deliberating over its possibilities. Right? So my other theme is science is slow. Science takes time. We are so worried about, was the data done last week correct or not? We really need to be looking over long time scales. So let's take this. Evelyn Gentry in 31 recognizes this vicarious trial and error behavior. Tolman suggests it's really about imagining the future, and everybody says rats don't do that. But these people are before Turing and Shannon, so they don't have concepts of information theory yet. They're before Norbert Wiener and John von Neumann, so they don't have computational complexity. They're before Alan Newell and Herb Simon, so they don't have the idea that data can be hidden within a representation. You have to work to pull that data out. They're before the 1960s, when neurophysiologists learned how to do single-cell recordings in awake-behaving animals. They're before 1971, when John O'Keefe got place cells and discovered place cells. Remember, Adam's observing place cells, right? It's before the 1990s, 1993, 94, when Bruce McNaughton and his postdoc, Matt Wilson, figured out how to record from a large neural ensemble of 150 cells. It's before the 1990s when computer people like me came in and we figured out, not just me, but lots of people, figured out how to do decoding of those neural ensembles. And it's before the 2000s when Adam and Jaden and I figured out how to do decoding at cognitive timescales. Wait a second, we're after all that. What Adam discovered was that Evelyn Gentry was completely correct back 80 years. This is an 80-year timeline to figure this out. Science is slow. So what we now know, and I'm going to skip over, because I'm only supposed to do 20 minutes and not 4 hours. I'm going to skip over a large amount of data and just tell you that we now know that rats and humans imagine and evaluate plans. The prefrontal cortex sends a message to the hippocampus that says, go create a simulated world. The hippocampus sends it to the nucleus accumbens, and the nucleus accumbens evaluates it, just as the nucleus accumbens evaluates the information that comes in the present. And I want to skip and go on because I want to tell you about the other half of what I've been doing here at the U for 20 years, almost. So I was sitting at the back of a very large auditorium, a 10,000-person auditorium, at Neurosciences one day, at the Society for Neuroscience meeting. And there was a researcher up front giving an addiction presentation. And this researcher, I won't say who it is, but this researcher made a prediction. And if that prediction were correct, it would have invalidated more than a decade of learning and memory research. That's cool. That's great. I'm actually super excited. This is the moment when I get to see a major theory get overturned and flipped over and wow, right? Except the next words out of this person's mouth was, we don't know why, but our prediction is backwards. And I'm sitting in the back going, what do you mean you don't know why? This is learning and memory 101. And so I wrote a paper, and I sent it off to Science, and suddenly, I call it my look over here paper. Kind of all you addiction people come look over what we've been doing in learning and memory, and my learning and memory colleagues are like, why didn't I write that? There's really nothing super special about it. But suddenly, I'm the addiction theory guy, and I get a call, among places, from NIDA, who calls up and says, why isn't our name on your science paper? To which the obvious answer is, because you haven't funded me yet, would you like to? So I have to say, NIDA has been very, very generous over the years since then, and has done wonderful things, both for my lab and for the U, and I'm very indebted to NIDA for their generous support. And they really have. Once this thing came out, suddenly I'm the addiction theory guy. So I'm going to take a slide or two and see if I can explain this insight. So in the learning and memory, in algorithms for how to learn things, there had been this idea called delta, which is basically how much better are things than you expected. So imagine we have some soda machine, and we'll assume it costs $1. I know soda machines cost more than $1 now. But you put $1 in your soda machine, and you get 2 sodas out. That's better than you expected, and you're going to increase your willingness to put money in that machine. If you put your money in the machine and no sodas, 0 comes out, you're going to decrease your willingness to put money in that machine because it's worse than you expected. On the other hand, if you put your money in the machine, it's $1, whatever it costs, and you get your 1 soda out, you still enjoy that soda just fine, right? This is not about pleasure or enjoyment. But in fact, you don't need to learn anything because you know how soda machines work, right? You're not changing anything. And there was a tremendous amount of work done in the 80s and 90s suggesting that this learning algorithm works really well. And in the 90s, that a brain chemical called dopamine tracked this signal. So Wolfram Schultz in the early 1990s had observed dopamine cells, and he observed them showing this very interesting property. And then I really want to make sure I give credit to Reed Montague, who in the mid-1990s recognized that what Wolfram Schultz had was this, what we call the delta signal. It's very important to give both the experimentalist and the theoretician credit. So so each of these lines is a recording from a cell, from a monkey. And you can see when you get the reward comes, an unexpected reward, you get a burst of spikes from this dopamine cell. Here, if the reward is predicted, then there's no change. And here, when the reward was predicted by a Q, so here this is a Q, when the reward is predicted by a Q but doesn't show up, you get a dip. This is better than expected, what you expected, and less than expected. And so I said, OK, let's walk off a cliff. Let's believe this theory. What if dopamine really is delta? If dopamine is delta, then we start from the hypothesis that dopamine is delta. And then we know that lots of drugs, not all of them, but many drugs of abuse produce dopamine. And what that means is that they will produce a better than expected. Even if you already don't feel pleasure from the drug anymore, the dopamine chemical says that was better than expected, do it again. And I basically did some very simple simulations to show that some of the addictive process can be caused by this dopamine signal. So OK, now we have this addiction as a dopamine process gone awry. Suddenly I'm the addiction theory guy. I told you about NIDA. But actually, one of the other things that happened is that the people here, again, the collaborators, the mentors, started latching on and saying, let's bring Dave into this. So Dorothy Hatsukami and Paul Pentel brought me in. Dorothy had a P50 center grant. And she said, I'm going to give you one of our pilot grant sub-grants. I hadn't applied for it. She said, I want to give it to you because you're going to still keep working on addiction whether you like it or not. And Ginger Siebold had a T32 training grant and said, I want you to be a mentor on this training grant, and I'll help pay for some of your grad students to keep working on addiction questions. But also, Ginger asked me to teach the theory day. Remember, suddenly I'm the theory guy. The theory day for their drug abuse addiction class, right? I don't know anything about addiction theory. So I started reading papers. At one point, I had my entire floor of my office covered in stacks about this high of papers reading the theory. And what I discovered is that all of these addiction theories said, if you have this problem, it might create an addiction. You might have an addiction. And all these other researchers would say, you're wrong. My addict doesn't have your problem. I hope everybody realizes that's backwards. If X then Y does not mean Y then X. And so I realized that all of these, actually not a lot, all of these if you have this problem things, were basically failure modes. They were errors in decision-making systems, and that addiction was a symptom, not a disease. And so we wrote a paper, and I like to think, and we brought in this idea of what's called a failure mode. Failure mode is a concept from reliability engineering that basically says, where's the vulnerability in the process? So for example, when the bridge collapsed, the 35W bridge collapsed about 10 years ago now, it doesn't collapse randomly. It collapses at a specific place because of a specific cause. It collapsed because the gusset plates were they were not built to spec, and then they were rusted out. Right? They had a failure mode for this bridge. And that leads us to this new concept called computational psychiatry. And the idea of computational psychiatry is that behavior is caused by lots and lots of different causes, like genetics and pharmacology and your environment, and because we're social animals, our social structure. But that these are all translated through the complexity of the brain. And what that means is if there's an underlying dysfunction somewhere, it creates a computational dysfunction that leads to the observed dysfunction. And so we could rethink psychiatry as computational dysfunctions in decision-making. So this is cool. The problem is, in order to do this, we need to understand how decision-making works before we can talk about what the failure modes are. Right? But I've been working on that. That's this other side. So we're not ready for computational psychiatry yet. But this is basically the other half of what we've been doing. And skipping over again about 4 hours worth of talk, what we know now is that we are not unitary decision makers. We are collections of multiple decision-making algorithms. And that these different decision-making algorithms access different brain circuits. So for example, the decision, is it worth it? That is, do I want to press this lever or not? Turns out to access a completely different neural circuit than which choice do I want to make. And similarly, what we've been doing now in a lot of our work in my lab has been looking at the difference between a foraging task, which says, how long should I wait for my cherry, or should I basically skip it and try my luck at another spot, versus choosing between two options. So this computational psychiatry idea means that we really want to translate from rats to humans. How do we get there? I started collaborating with Angus MacDonald, who's a professor in psychology, and Angus and I co-advised a graduate student named Samantha Abram. And Sam has graduated recently. She's now a clinical fellow at UCSF. She was a clinical psychology grad student. And Sam set out to try to do this translation. And then at one point, we were really having trouble kind of figuring out how to do this. And at one point, Sam comes into my office and says, I've got it. I know rats forage for food. I know what humans forage for. And I said, what? And she said, kitten videos. And so we created a foraging task for humans where you go up and you basically ask, is it worth waiting for my kitten video, or do I skip it and try the landscape restaurant next door? And now mice, rats, and humans all look identical to several orders of magnitude. And I want to point out this big science paper, which is the sunk cost paper that Dr. Toller was mentioning, that this is actually the collaboration of 3 different laboratories here, my laboratory doing rats, Mark Thomas's laboratory doing mice, and Angus MacDonald's laboratory doing humans. And that this took us over 5 years to actually get everything aligned. Tremendous amount of work with several students and lots and lots and lots of help. So, but now we have this kind of neuroeconomic side, and that means we can start to talk about computational psychiatry. And computational psychiatry took a big step in 2015 when I organized a meeting called a Strungman Forum. Now, a Strungman Forum is a very strange meeting, very specialized meeting, where you bring in 44 senior scientists, not junior people, but senior people, and you lock them in a room in the suburbs of Frankfurt and you make them talk to each other. Now, you're actually going to write a book together, but you have to get the image. These are not junior people with like really flexible ideas. These are senior people who have been working on something for 40 years and know exactly what they're talking about. They have lots of data in the background, and they've got big egos, and you've got to get them all to write a book chapter that they will agree to sign their name to. This turns out to actually be incredibly useful. And it was so the problem was, I should say, before this, I knew the computational people. I could read people like Reid Montague and Peter Diane and the people who do computation. I didn't know any psychiatrists, right? I'm not a clinical person. I'm not medically trained at all. I'm a computer jock, right? But I had a friend, a guy named Josh Gordon, and Josh and I go way back because he's a place cell guy working on mouse hippocampus mouse place cells. And I was like, Josh, don't you have a side gig as a psychiatrist? Could we co-do this and you bring the psychiatry people in? And he said, yeah, I can do that. And so we organized this meeting. It was by far the most dynamic, amazing, incredible meeting I've ever been to. And I actually called my wife when I came after the meeting saying, I think we just changed the world. This meeting changes how we should think about psychiatry. And again, it wasn't me. It was this dynamic meeting of these people in this organization, this incredible thing. Now, let's be honest. This is a bunch of scientists. It's not like NIH is going to change. It's not like the University of Minnesota Psychiatry Department is going to change clinical intake or anything like that, right? This is just a bunch of people talking. Well, about 6 months after this meeting, Josh calls me up and says, Dave, I'm going to take the job. And so some of you may be saying, I know that name Josh Gordon. Why do I know that name Josh Gordon? Because this is the Josh I know, and this is the Josh you know. And the job he said he was going to take was the director of NIMH. And so NIMH has gone all in on computational psychiatry because this little book we edited suddenly became a lot more important when my co-editor is now director of NIMH. And then something else happened, which is that the University of Minnesota hired this absolutely amazing dynamo of a woman who showed up, and I was not part of the hiring decision. I wasn't involved at all. And yet when she came here, she was talking about what's the blood test for psychiatry. And everybody said, you sound like Dave. Have you talked to Dave? So I came over and she invited me over to come give a grand rounds. I gave a grand rounds. And I have to say the psychiatry department was incredibly friendly and really involved and really kind of ate it up. And then the two of us were sitting in her office afterwards, and things just clicked. And the next thing I know, we have building on some neuroscience advisory board that she'd started to build. We ended up co-facilitating this thing we call NeuroPrism, which is a group of faculty that have met, I think it's for 2 years now. Are we actually at 2 years now? That 30 faculty come and have met every other week. And those of you who are faculty know how crazy and hard that is, because we're not telling them to do that. We just said you can come if you want. And these 30 faculty have come every other week, and they're discussing and they're changing. And so I should say it's about half clinicians and clinical scientists, physician scientists, doing clinical work, running the actual clinics in things like, working with anorexia, working with OCD, working with first psychosis, and the basic scientists, the fundamental scientists, working on rats, mice, monkeys, and humans, but doing the fundamental science questions. And grants have come out of this, and papers have come out of this, and they're now actually changing how they do the clinics based on these. There's translation from the rats and mice to the clinic, and honestly, from the clinic back to the rats and mice. There are questions we didn't know were questions until we did that, right? Now that we have this going back and forth, it's this incredible dynamo. I feel like I'm riding a bull by the horns, right? But it's a wonderful thing. And so I'm going to end there and thank a few people before I finish. And I want to obviously thank external funding sources, but I also want to take a moment to thank internal funding sources, because one of the things that the university has done that's been tremendous over the years is that they've essentially caught me. There's a death wall. Some of you who are not faculty don't really know this, but if your funding really runs out and your lab shuts down, it's very hard to restart. And they've caught me several times as I've hit that, and I'm deeply indebted at the willingness of the U to kind of keep us going. I also want to thank all of the undergrads who volunteered their time to come work for us, the grad students and postdocs who come in and, of course, done most of this work, the rats. Don't forget the rats. I want to end, though, by thanking 3 people, because without them, I couldn't do what I do. Ayaka Sheehan, Chris Bolt, and Kelsey Seeland. Ayaka's been with us for about 5 years. These are the technicians who run the lab. Chris and Kelsey came in actually as undergraduates in fall of 2000, and they've been with the lab for the entire time. This lab is as much theirs as it is mine, and I couldn't do this without them. So I'm going to end there, and thank you very much.
Good afternoon. I'd actually like to start by inviting you to join me in my clinic. We're sitting in my office. I'm evaluating a new patient. Her name is Sally Johnson. She's 21 years old. She's a student here. And she's come in because she's become increasingly withdrawn. She's irritable. She's accusatory of the people around her. She's not sleeping very well. Her roommates have become quite concerned because yesterday they found a list in her room where she's written down the names of people who she thinks might be trying to kill her. It's 2018, so we know that these kinds of symptoms that Sally is experiencing, they are the product of our brain, just like all of our subjective experiences, our thoughts, our feelings, our emotions. Those are the output of our brain. And yet, as I sit there and I listen to Sally, as I talk to her, as I try to evaluate her, the only tool I have at my disposal to try and figure out about what is happening in her brain is observing her, asking questions, listening to the answers that she might choose to give me. I have no way, no lab test, no means of actually determining what is not working in Sally's brain. I don't have a way of pinpointing the exact dysfunction, the exact failure, to use David's words, and of correctly prescribing a specific treatment that will correct her brain dysfunction. Unlike any other branch of medicine where the assessment, the evaluation, the decisions about treatment, they're informed by an understanding of the physiology of the organ. As a psychiatrist, I don't have any of that. At the best, I can make a rough guess based on the observations that I have, and then I can prescribe usually a medication. And the medications, though, they are very useful because they alleviate symptoms. As Dean Tolar suggested, they are in a sense palliating. They're not, again, correcting the underlying brain dysfunction. So I would like to tell you about this journey that I've taken to try and change that situation, to try and bring science into the practice of psychiatry, and the story of how I got there, similar to the story, Dave's story, about how he got to be standing here in front of you today. And it's a story not just of serendipity and collaboration, but also of a little bit of stumbling also. The story starts, these are my disclosures, because I'm going to be talking to you about some research where I've had different kinds of support and potential conflicts. But I'm going to tell you about the beginning of the story. And the story actually starts when I was a resident, and I was about to begin my residency at Stanford. And this is where the serendipity begins. I was actually offered the psychiatry residency position when a prior applicant backed out. They had already set up a residency class that had 5 MD PhDs. They all happened to be white males. And I think when this one person backed out, they said, you know what, we need to diversify a little bit. I was there doing a rotation, so they asked me to join. I think they thought I was going to be the closet clinician. There had to be somebody taking care of the patients while everyone else was out in the lab. As a resident, I became interested in working with people who had psychosis. So psychosis is a term we use to describe a loss of touch with reality. Hallucinations, for example, delusions, believing things that aren't necessarily true. And I was very struck by how is it that a person can come to believe very deeply in their bones that, for example, the FBI is trying to control them with radio waves, or that they have become the chosen person who's going to unlock the secrets of extraterrestrial communications on this earth. How does that come to be? So at the time, we were taught as residents that schizophrenia is caused by an excess of dopamine in the brain, and that the best treatment would be medications that block that excess dopamine. But I was struck by something else as I listened to my patients, as I observed them. I was very struck by a certain pattern that I kept seeing, a certain sort of formalism. And that pattern was that patients inevitably started to set up these complex meaning structures, these interpretations, to pull together what were random sensory perceptions. So an example might be someone coughed, a door closed, a truck went by. And for us, our brains just sort of let that be. That's random. But when people were developing delusions, particularly in the early stages, all of these things began to be tied together. They began to have meaning, and meaning was being ascribed to them, often meaning that had sort of a threatening component, but not always. I decided I wanted to do a biological psychiatry fellowship because I wanted to learn more about schizophrenia. But on the side, something serendipitous happened. I got a chance to work with Irv Yalom, who was a faculty member at Stanford. We were on, when I was rotating through the inpatient unit, he was the attending. And Irv turns out to be this incredibly talented psychotherapist, as well as someone who is an existentialist, a novelist also. He's written a number of novels. But as a group psychotherapist and working with him as a trainee, learning about group psychotherapy, Irv taught me that even though psychiatric illnesses might essentially be failures of decision-making, failures of the subjective experience, that inevitably at some point they would lead to failures in interpersonal interactions, failures in how the self interacted with the outside world. So it's another kind of failure mode. That was important. He also taught me that these failure modes could be ameliorated. These dysfunctions could get better if people could learn new skills. So that's the idea of brain plasticity, that these systems are not fixed, but that they're changeable. And then also that sometimes people might need to learn to compensate, just develop skills that help them compensate, this idea of sort of compensatory mechanisms. So this was a very exciting time. I was sort of balancing between sort of the biological realm, where I was sort of getting excited and thinking about new projects, and then this more psychological realm, which was deeply clinically meaningful. And then I would say a key stumble happened. It turned out that some of the people that I was working with as my mentors in my biological psychiatry fellowship, they really, in some way, were anti-mentors. This was a particular time and place. The department was going through a lot. There were particular people who were in positions of power. And as a fellow, senior resident as a fellow, I ended up working with a team of people where, at one level, some of my ideas and interests were sort of dismissed. They weren't perhaps deeply biological enough. I was left out of certain projects and papers. And there were also a couple of incidents which I think in today's day and age we would call sexual harassment. And I have to say I've never actually talked about that publicly before, but it seems appropriate because I'd like to be able to, in a sense, model what happens after this kind of an incident. What happened to me is I lost my sense of confidence, and to a certain extent my sense of purpose. And the stumble, I left academia. I decided to take a position in San Francisco, a completely community-based position in a community hospital, running an inpatient geriatric psychiatry unit. There I was at the beginning of my career, having lots of these ideas and interests I've described to you, and in complete scientific exile, in a place where really the pursuit of some of these ideas I'd been getting excited about was really not the time or place. But there was still more serendipity in store for me. I had the good fortune of being friends with Roy King. He was a mathematician turned psychiatrist who was on faculty at Stanford. And while we never worked directly together, we stayed friends. And we were having coffee one day, and I was beginning to describe more of my interests in sort of the development of paranoid delusions. And Roy told me about Bernardo Humerbin, who was a physicist and information scientist with whom he had been collaborating. And here comes some of the first collaborations. We all got together, and I started talking to Bernardo now, as well as Roy, about these observations I'd made as people are developing paranoid delusions. And these observations about this creation of these networks of meaning that are happening from these random events. And Bernardo says, spreading activation, no, I'm not even going to remember what he said, phase transitions and spreading activation networks. I'm a little bit emotional talking about this point in time. But we have this conversation. Bernardo says, you know, I've been modeling things which really sort of fit in with this. Let's write a paper. So we wrote a paper about that, and that was in 1992. And I've not even told you about this paper. But it was like my first step into that computational domain. At the same time, in this community hospital, I was teaching myself some aspects of essentially how to perform psychology experiments, cognitive psychology experiments, because I was kind of getting that aha moment of not only can mathematical models of brain information flow actually provide these really satisfying explanations for what I was seeing clinically, but that also brain information flow could be inferred from doing these kinds of experimental tasks in patients. And then another serendipitous thing happened. Because I'd worked with Irv Yalom, I got invited to apply for a faculty position at the San Francisco VA after a failed search. The prior applicant had backed out. There were no women faculty at the VA at that time. They wanted to hire a woman. I got invited to come and interview by a senior colleague who had worked with Irv and wanted to start a group therapy program and said I would be just the person to do it. Little did they know that I had all these other interests developing in terms of psychosis and delusions and mathematical modeling and so forth, but I accepted the position. I have to say, not too long after I accepted this position, I had set up a rotation for medical students to come and work with me in clinic, and we developed a clinic focused specifically on schizophrenia and focused particularly on biological aspects of schizophrenia, and one of my medical students was Josh Gordon. I can claim to have taught him everything that he knows. Around this time, because of my publications on psychosis, because of my clinic that I created focused on schizophrenia, I was invited to join the anti-death penalty team that was working on the case of Ted Kaczynski. Some of you may know of this case. Ted Kaczynski was also called the Unabomber. And this was a front row seat into a number of things, including how delusions develop over time. Ted had kept extremely extensive journals dating from his years in college all the way up until the time he was arrested. And in these journals, he chronicled some of his internal processes as well as kind of the conclusions he was trying to make about what was happening to him. And when I was able to sort of watch unfold over time what started out someone who was a brilliant mathematician, he had actually been on the faculty at the University of Berkeley, was beginning to have social difficulties, interpersonal difficulties, concerns about how people might be interested in the work he was doing, concerns about societal control over his mental processes, and to watch that process go from a functioning mathematician to someone who, a couple of decades later, ended up leading this reclusive life all alone in Montana and being someone who carried out serial homicides because of his delusional thinking. And so this got me thinking about what is that trajectory? What could one have done differently to interrupt this trajectory? Some of you may know that thanks to the hard work of the defense team, we were able to make a case that instead of being given death, he'd be given a sentence of life without possibility of parole. I'm now a junior faculty member at UCSF. I'm continuing my interest in psychosis. I'm doing some experiments, trying to understand information processing in psychosis. And one day, I go and listen to a seminar being given by Michael Merznick. Michael Merznick is a basic neuroscientist. It's around 2000 now. And here's a second light bulb going off. Because what Mike talks about and the language he uses just is this revelation for me. It's this notion that the brain is representing and processing information through the coordinated activity of its neurons, the cells in the brain, across lower and higher level systems of the brain, systems that take in information, systems that make sense of information. And then, of course, Mike, this is what he's known for, is making the point that the brain is characterized throughout the lifespan by significant amounts of plasticity. The brain is, by definition, an organ that is continuously changing and remodeling itself. So for me, the aha was, you know what? Psychiatrists really need to start thinking like systems neuroscientists. Systems neuroscience is the basic science of psychiatry. And what I mean by that, quite simply, is that it's this notion that you've got brain information processes which are flowing through brain circuits. And this flow of information, that's kind of what is making us be who we are, be the human beings that we are with all the things that we're able to do. And this proper information flow has to be coordinated and integrated just the way a symphony has to coordinate and integrate all the different elements that are happening in the orchestra. So you've got different sections that are playing different parts, in a sense, if you will, of the musical score, different parts of the brain, different regions, different networks, different circuits. You heard David describe some of them to you. And you've got a conductor who is kind of interpreting, integrating, bringing all the pieces together to create, integrating it to create the experience, the behavior, the output. So I'd like to sort of make this more real for you by using a metaphor, an actual metaphor of a symphony orchestra. So we're going to listen to how it works so well when all of the pieces are coming together. And now, when it's not working so well. Same musical score, same instruments, but a few things are just a little bit off. Right? So this is a very nice metaphor for what is happening in psychiatric illness. The neurons are still there, the brain circuits are still there, the instruments, if you will, are still there, but there's a few things which are just off in terms of how things are being represented, in terms of the final output. Perhaps the conductor is a little bit immature, perhaps the prefrontal region of the brain isn't fully mature, perhaps not functioning properly, perhaps some of the sections are out of tune or not coordinating well with the other sections. This is the metaphor for psychiatric illness. So in collaboration with Sri Nagarajan, who is a biomedical engineer, we set out to do a number of experiments using different imaging techniques to probe the ways in which the brain information flow was out of tune in psychosis, in illnesses such as schizophrenia. And here's a nice example from very simple information processing in the auditory system. This is an individual listening to 2 syllables. This work was done by Corby Dale and Alex Herman. Alex Herman when he was a medical student at UCSF, and I'm very proud to say he came out here and is now a research fellow here in the MinDrive Neuromodulation Fellowship. But here's how a healthy brain, this is the flow of information in this region of the brain over time. This is what we call high gamma neural oscillations. And here we have the healthy subject, the flow, ebb and flow of the information in this part of the brain. And here's the person with schizophrenia. And you can say that even in the simple listening to 2 syllables task, the brain is not, the information flow through the brain, the representation of information is abnormal. But I was also interested, this was influenced by my work with Irv Yalom, no doubt, in higher order functions, functions that have to do with how the self is relating to the world. It's something that we sometimes experimentally call reality monitoring. And I'd like to dig a little bit more into this experiment with you because I think it reveals a lot of interesting information about what goes wrong in psychosis. So in this experiment, a reality monitoring experiment, what we're going to do, we're going to pretend you're one of my research subjects. And before we take you into the scanner, I'm going to show you this, we're going to do the study phase. I'm going to show you these sentences. And you can see that some of the sentences, there's just a blank word. I'm going to ask you to make up a word. It's going to be self-generated information, or I'm showing you a word. I, as the experimenter, am giving you the information that's coming from outside of you. So you might read, the rabbit ate the carrot is fine. We'd record that. There's no right or wrong answer, believe me. The sailor sailed the sea, right? Dog chased the cat, whatever you'd like, right? Now you're in this, that's fine too. Just shows your creativity. Now you're in the scanner. And while you're in the scanner, I'm going to ask you to make a decision. And the decision is a reality monitoring decision in which you need to distinguish between information that you generated yourself versus information that I had presented to you. So it's a self-other distinction, if you will. This is normally very, very easy for people to do. So you see rabbit carrot, and you say, I made it up. It was self-generated. You see sailor sea. I read it. It was generated on the outside. Fine. So we now take a picture of your brain as you're doing this task. We look at your behavior. What we see, this is the performance of healthy people, what we see is that when people have a psychotic illness such as schizophrenia, this task is more difficult. They cannot distinguish between information that they have generated within themselves and information that has actually just been presented to them in the outside world. That makes sense for you when you think about the symptoms of schizophrenia, hallucinations, delusions, disorganized behavior. But they also show a different pattern of brain activation when they're doing this task. Healthy people activate this medial prefrontal region of the brain here. That's a region of the brain very important for the processing of self-referential information, also very important in the social self. People with schizophrenia are activating a posterior region of the brain. They're using, in a sense, a different brain network as they try to do this task. Again, the information flow in the brain is abnormal, and it's abnormal in ways that have to do with even these subtler, higher order, self-other kinds of distinctions. So of course the question, and this came from now working with Mike Merznick for several years, is can we use plasticity processes in the brain, this natural, normal set of processes that the brain has to continuously remodel itself, to figure out ways to retune this broken symphony orchestra, to get more adaptive information flow happening through these circuits. And so we set out to develop a series of training exercises, cognitive training exercise, focused on different circuits in the brain in an attempt to do that. I'd like to show you a couple of examples. We're going to start with an example of the auditory system. Let's see if this is going to work. So this is a very, let's see, there we go. Very easy, one of the tasks, 2 sweeps. The brain has to figure sweep going up, sweep going down, and then respond correctly. This is an easy level of the task. It gets more and more difficult as people are able to do it. If you had schizophrenia, even this level would be a little bit difficult. Now we're going to look at some training, where we attempt to train up some of these higher order social cognition functions. In this case, the ability of the brain to quickly register, hold online facial information, and then make a correct response around it. Processing facial information is very difficult for people with schizophrenia, as it is also for people with autism spectrum disorders. And you can see, as the task is getting slightly more difficult, faces are rotating and so forth. So this is an example of the kinds of training we did. We did 16 weeks of very intensive training of the auditory system, the visual system, and this social cognition system. And what we saw is after 16 weeks of training, individuals with schizophrenia were showing a couple of changes. The first change is that they were now able to do this reality monitoring task with a greater degree of accuracy. And so this is before training over on the extreme left-hand side. This is after training. They've improved task performance. They can now make these self-other distinctions in a way that is more closer to healthy people than they could before. We're now seeing activation in this medial prefrontal cortical region of the brain. So they're now bringing online that part of the brain that they were not using before. We're seeing this is the signal change in that part of the brain in the people who got the training. You can see that the active training group, you can see it significantly increased. This is a computer games control group. This is a healthy comparison group. All of the assessments were done blinded, I should say, goes without saying. And we now see that the signal in the medial prefrontal cortex is correlated with task performance. They're now using, the Symphony is now in tune, they're now using the Symphony in the correct way to accomplish this reality monitoring task. This work was done by Karuna Subramaniam, who was a postdoc in my lab. Perhaps more importantly, because at some level when you do this kind of work, you can say, well, so what? So this brain activation looks more normal, so they can do this laboratory task in a better way. What does that really mean for people in the real world? Where more importantly, what Karuna showed in this paper is that the change in prefrontal cortical activity that we observed immediately after training, so the gain in that signal correlated with real-world social functioning as we assessed it 6 months later. So those people who could mount this increased activation in the prefrontal cortex after the training were those people who showed the most change in their real-world social functioning. Well, this was exciting for us, but then, and I think in part stimulated by the work that I had done several years previously on the Ted Kaczynski case, the light bulb went off again. And here the light bulb was, well, this is one thing to do this in people who've been ill on average for 20 years, because we were studying people who had established illness who were in their early 40s. But what if we tried to intervene very early? At that phase, using the example of Ted Kaczynski, when he was just beginning to think that perhaps he was being followed, or perhaps there were things happening to him for a reason, not at the stage when you have full-blown delusions. What if we thought about intervening in a way to restore brain circuit functioning, to preempt the deterioration, either before the onset of the disease or at least around the time of the initial diagnosis? And this led us to carry out a multi-site study with colleagues at UC Davis. And I particularly want to thank Melissa Fisher, who's here today with her husband, Jeff. Melissa has been working in my lab for a number of years. Again, one of those people without whom I could not do this work. She was willing to move to the University of Minnesota. She's an associate professor in our department. And her husband, Jeff, particularly also gets a thanks for being willing to do the move. And she played a lead role in this particular multi-site project. But what we did is we now worked with young individuals who were on average 21 years old, who'd at most been ill for a couple of years. And we did training just focused on the auditory system, very low-level training to tune up the abnormal information processing in the brain. And we looked at changes immediately after training, but what I want to show you again now are the changes we saw 6 months later after we finished the training. And what we saw is that, first of all, the group that got the training in blue showed significantly better cognition on a range of pencil and paper tasks after they got the training compared to a computer games control group. But more interestingly, this group showed lower symptoms. So their symptoms decreased, this is symptom ratings, over time. 6 months after the intervention, their symptoms are significantly lower than the group that got the computer games control condition. This is very interesting because there was nothing about what we were doing that was actually targeting symptoms per se. We weren't blocking that dopamine in the brain with dopamine blocking medications or anything like that. We were retuning the symphony to play in a much more harmonious and adaptive fashion, if you will. Ian Ramsey, who's now a postdoc in my lab here, went on to look at some of the brain changes that we saw in these young individuals. Among other things, he's shown that there is a thalamic volume, increased thalamic volume is associated with increased cognition in the group that gets the active training in this slide in red compared to the computer games control group. Ian has also shown that there's changes in connectivity between the thalamus and the cortex in the group that gets the training, and these increases are associated with changes in cognition. Interestingly, this change seems to be compensatory. The brain in people with schizophrenia is using a network that healthy people would not use to be able to sort of get this better cognition, but it's working for them. It's a successful compensatory strategy. So this body of work was well underway, much of it completed, when about 2 years ago, I left San Francisco to come and join the faculty here at the University of Minnesota. And you might say, well, why? Why would you leave San Francisco to come to Minnesota? Even though there is this great bridge here. And I think there's several answers I'd like to give to that question, and they're answers that fit very, very well with what David has been talking about, what Dean Tolar has been talking about. And I can start with a conversation that I had when I was being recruited here with these three people. Kelvin Lim is in our Department of Psychiatry. He's a very well-known professor, known around the world for his neuroimaging work. He's an engineer turned psychiatrist. I was actually a chief resident of Kelvin at Stanford, so I can say I taught him everything that he knows as well. But Kelvin arranged a conversation with Angus MacDonald. You've already heard about Angus from David's talk. Angus is here. By the way, Angus gets credit for the acronym NeuroPrism, which you didn't tell people about, which is Neuroplasticity Research in Support of Mental Health. But so Angus was at this conversation, and then a basic neuroscientist, Matt Chaffee, was there, and Matt does work in animal models of abnormal information processing flow relevant to psychosis, and Angus, you've heard about his work. And we all sat in the room, and we talked about the research we do and the focus we have, and I was just blown away that these kinds of active collaborations across departments and specialties and disciplines was happening, and it was happening in a way that was directly relevant to the things that we need to tackle in psychiatry. At that same visit, I also had the chance to have conversations with Camille Ugerbil, who runs the Center for Magnetic Resonance Research, played a major international role in the Human Brain Connectome project, and Konstantin Aliferis at the Institute of Health Informatics, who's very, very interested in computational psychiatry and has worked with me to hire faculty who bridge that informatics psychiatry divide, including Jessica Nilsson, who I think I saw is also here, who came from the University of California, San Francisco to join this endeavor. So I had these conversations, and it was clear to me that there was these incredibly deep resources here that could help directly address what I felt needed to happen next in the field of psychiatry. And then, of course, I knew that Dave Redish was here. Even though we didn't meet during my recruitment visits, is true, we met not too long thereafter as we began to create these groups where the neuroscientists and the psychiatrists, the psychiatric researchers began to get together. And I think it's safe to say that we embarked on a series of both conversations and work group activities together that for me have been transformative both in terms of how I think as a scientist, but also as a psychiatrist, and what I now feel we can accomplish. So it's safe to say that here at the University of Minnesota, it was clear to me when I was being recruited, but it's only become increasingly clear to me that what we have here is this incredible cadre of neuroscientists, data science colleagues, imaging colleagues who are focused on these aspects of brain circuit dysfunction, of neuroplasticity, of mathematical modeling, all of these things which were these light bulb moments over the path of my career, which has led us to create this NeuroPrism group, Neuroplasticity Research in Support of Mental Health. But I want to mention two other, in a sense, things that are here at the University of Minnesota that are quite compelling and are not necessarily found at other institutions. I've been able to have collaborations with a woman named Piper Meyer-Kalos in the School of Social Work, who is nationally known for her first episode of psychosis clinical program development work around the country. And she has been incredibly collaborative, and we've been setting up new first episode of psychosis clinics. Now we have 4 of them in the state, and they've been funded by the state, and that's thanks to, in large part, to Piper's work, as well as Sue Abderholden, who's the executive director of NAMI, which is the patient advocacy group. And the vision and drive of people such as this has made it clear that what is also here is a set of community collaborators who are really ready to bring science and brain plasticity-based approaches out into the field, not just even into the university clinics, but out into real-world treatment settings. But it's also important to remember a third component, a third kind of leg of the tripod, if you will. I was fortunate enough to be introduced to Lynn and Andy Redleaf, and then to have some really remarkable conversations with them. They're also here this evening. And based on those conversations with them, as well as with Wendy Wells of the Wells Foundation, it became clear that what is also here are donors, philanthropists, who are willing, who have the vision and the willingness to support high-risk projects that are going to help bring science into the practice of psychiatry, because this is not something that traditional funding agencies are going to be undertaking quite yet, not the direct translation into practice. With these amazing collaborators, and I've just shown you a few of them on the previous slide, I now very strongly feel that we are ready here to accomplish this quite exciting challenge of bringing science into the clinic. And again, with the support of donors, with the support of the science colleagues, we're thinking about ways of creating tools that will allow me, when I sit and need to try and evaluate a patient as Sally Johnson, to be able to actually map what is happening in the brain, to actually come up with a much more precise approach to making treatment recommendations. And I actually think that we're going to be able to do this in a way which is a model nationally and perhaps even internationally. And I actually think it's just a few years away when, the patient will walk into my office and I'll have something more to offer than just the observations and the questions that I can ask, and perhaps a prescription for a medication. So I'd like to thank you for your interest. I would like to thank the people who have funded our work. And of course, I'd like to thank our research participants and their family members.